And our last speaker today is Erin, I'm going to get this one wrong too. Yaglova? Lagacholova. Okay, and the title of her presentation is Enhanced Microbial Methane Oxidation in Landfill Cover Soils. Let's see. Do you have a pocket?
I don't actually have a space.
Okay, maybe attach this right
To the oh, so it's okay. Thank you. So my talk is about enhanced microbial methane oxidation in landfill cover soils. So I'll be going over first what the problem is and the objectives of our studies, which include details on landfill gas generation and the associated greenhouse gas emissions, and the potential for biochar amended biocovers for landfill methane mitigation. Then I'll go over some experimental studies at UIC that were performed by a PhD student who has since graduated, Pupak Yagubi, which she conducted long-term column incubation studies and batch experiments to determine the kinetic parameters of methane oxidation. And then I'll conclude with ongoing studies that I'll be performing at UIC and some future work and conclusions. So what is the problem? As many of you might have heard, especially from our previous presentation by Yamini, that landfills are a source of greenhouse gas emissions and are actually the third largest source of anthropogenic methane in the United States. So they contribute a significant amount of methane over their lifetime, which for some landfills can be, decades. So it's estimated that 500 to 800 megatons of CO2 equivalent are released from landfills globally per year. And with increases in population, these numbers are expected to continue to rise. So the problem is not going away anytime soon. So we're looking for a effective way to deal with these emissions in the long term. Many new landfills have landfill gas recovery, which does help mitigate some of that methane generated. But a lot of older landfills do not have those systems, or those systems are not operated all the time because they need a certain minimum level of methane to be economically efficient. So there are a lot of areas for improvement that we can use biocovers for. So the IPCC had identified biocovers, which I'll explain more about what those actually are, as a key mitigation strategy for this landfill gas. Oh, yeah, so we need some sort of practical, economic, and effective long-term solution, as I had said. Okay. So shown here is basically a graph showing the lifespan of a landfill and the amount of methane that's generated over that lifespan. So typically, you'll see after closure, during construction actually still going on, you'll see increases in methane generation to a peak, which will gradually decrease. But as you can see by looking on the x-axis, that can go on for decades. So the amount of methane that's not captured, which is illustrated by the difference in recovery rates and methane emissions, that's all the methane that we're trying to target and mitigate with these additional biocovers, which would go on the surface. And above that, you'll see an equation. Just briefly, methanotrophs are bacteria in soils that are basically ubiquitous at low levels throughout soils. They're typically enriched in areas where there's methane emissions. So these bacteria use methane as their sole carbon energy source and oxidize it in the presence of oxygen, forming CO2 and water. So those are actually the mechanism that we will use or exploit to mitigate landfill methane. Oh, yeah. So just to point out, this is the methane not capture that we're focusing on. And a lot of that occurs in the operation phase during active landfilling and during landfill closure. So current biocover technologies that have been employed before have used a variety of materials. So the whole goal of these biocovers is to promote the growth of methanotrophs, those methane oxidizing bacteria, to oxidize methane into CO2. So previous biocovers have used compost, sewage sludge, mulch, peat moss, activated carbon, corn stover, etc. Any types of organic amendments usually are working. But what we found is, or what other people have found, is that the oxidation efficiencies of these types of biocovers are limited by a lot of several factors, including material degradation, especially in these labeled carbon sources such as compost or mulch or something like that. You can also, in some cases, get methane generation rather than oxidation if the water holding capacity is too high, and also coupled with the presence of labeled carbon, you could actually get methanogenesis during wet periods so in fresh compost or with labeled organic matter. There's another issue that involves the formation of EPS, or exopolysaccharide substances. These are substances that are secreted by bacteria during their metabolic processes that will basically coat soil particles and reduce gas transport and aeration through soil. So because methanotrophs need oxygen to oxidize the methane, reduce gas transport from either water or the production of these exopolysaccharide substances can reduce overall oxidation efficiency. So that's another concern in some of these biocovers. And furthermore, if you have a lot of ammonia, you can actually get inhibition of methanotrophic activity or competition with heterotrophs for nutrients. So there's a lot of issues that need to be resolved. So one proposal is to look at biochar as a substrate for cover amendment, because we need basically a superior material that is able to sustain methane oxidation for longer. It's not going to degrade or be too labile such that there's excess carbon, generation of EPS, and reduction of aeration. So biochar might be that kind of material. So just to illustrate the different types of ways that this could be applied on a landfill, there's several designs: biofilters, biocovers, biowindows. They basically differ in their construction. So whether it's, biocover would be covering, a large area. Biowindows maybe would be placed just at hotspots, or we call them hotspots, but places where there's higher landfill gas fluxes than other places, such as along sides or in cracks. Most of these include a gas distribution layer of coarse gravel at the base, as shown there, which allows the gas, it basically improves the uniformity of diffusion of gas through the soil. And also allows for like an increased retention time in the cover. And then above that, there's a low permeability cover placed over the waste layer. So generally, the reason they want it to be low permeability is to reduce, the gas transport out of the landfill. But as we'll see, or as I'll talk about, that might be a negative impact in terms of biocovers because of reduced aeration. So one way to apply biochar is either to spread it across in a thin layer or to mix it into the soil for one of these biocovers, and that would provide a carbon source and also a habitat for methanotrophic bacteria to colonize. So now I'm going to discuss the experimental study conducted by Prupak Yagubi that I mentioned earlier and what some of those results are. So the biochar that was used in the study was produced by Chip Energy, and we used it as is in the pelleted form. And soil that was also used came from the Carlinville landfill cap. It was a silty clay soil. So here's a schematic of the column setup. So what the goal was to simulate landfill conditions by pumping in synthetic landfill gas at the bottom here, having a gas distribution layer of gravel, about 15 centimeters, and then soil amended with biochar on top of that. And there's sampling ports along the depth of the column, so we can sample the pore gas at different depths, and as well as at the headspace outlet to compare the methane concentration in the outlet versus different depths and in the inlet. And from that, we can calculate oxidation efficiency. So the column was flowing for 4 months, during which there was daily measurement of headspace concentration and also along the depth profile. And after the 4 months, the column was extruded, and it was divided into 3 portions: upper portion, middle portion, and bottom portion, from which DNA was extracted for molecular analyses. Also, batch testing. So soil was taken in addition to that taken for the DNA extraction for batch testing experiments to determine kinetic parameters. And isotopic analysis was also conducted to infer microbial fractionation of methane. So here's a picture of the actual column. So there's 2 columns. One was a control, soil only. The second was biochar amended. So there's a 20% biochar amendment, 80% soil. Adsorption was assumed negligible in this case because measurements weren't started until a steady state oxidation was reached, and at which point we assumed adsorption capacity was reached. So here is shown how methane oxidation efficiency was calculated. So to use something called the fractional conversion of methane to estimate that oxidation rates. So it's just based on the differences in CO2 and methane concentrations. You only need to measure CO2 and methane. And there are some limits of this. It doesn't account for all the losses, potential losses of methane, but it's a handy way to estimate oxidation efficiency in the absence of measurements of oxygen. So what was found for the effect of methane influx on oxidation efficiency? So over the course of the experiment, methane influx was varied after steady state oxidation was reached, and measurements were taken to determine how that affected oxidation efficiency. So on the left, column 1, the unamended column, so soil only, and on the right, column 2 is the biochar amended column. And from here we see that at all flow rates, it's lowest on the left going to increasingly higher, at all flow rates, biochar amended column had a significantly more methane oxidation. So that was a positive result. You do notice, though, in both columns, there is a decrease in oxidation efficiency with increasing methane loads. So that indicates there's some sort of maximum capacity that the methanotrophs have. They can only oxidize so much methane. If you give them too much, maybe in a while they'll get used to it, but immediately after they are not able to quickly ramp up their oxidation efficiency. So these findings are consistent with other landfill biocover studies with different types of substrates. So this was a promising result. And just to note, even though it's a relatively low efficiency, it's nearly 20% at the lowest flow rate, that can still amount to significant methane oxidation over time. So considering a lot of landfills don't have these biocovers to mitigate methane emissions, this is still, I think, a promising result. So here is shown a gas profile. So we have both, actually this is just methane concentration, but as you can see, this legend is kind of complicated, and I apologize, but the closed circles, or closed shapes, are all column 1, the unamended column, at different flow rates. The open shapes are the biochar amended column at different flow rates. So you can kind of just look at the open shapes and the closed shapes. So from this, we just generally see more methane oxidation or lower methane concentrations in the biochar amended cover. And also, if you just look at the depth to which those reductions go to, you see a greater depth of oxidation. So the oxidation front is moved down in the presence of biochar. Because there is actually an oxidation that will occur naturally in soils, as observed in the control. You can see some methane concentration is decreased in the surface, upper 10 centimeters, but that's basically restricted to the upper 10 centimeters. The biochar is affording a greater depth of oxidation zone, so more oxidation in general. So, but past this oxidation horizon, the effect is negligible between the two columns, and at all flow rates, you basically see the same methane concentration. So at 2 points in the experiment, there was moisture added to simulate a precipitation event, and gas profiles were taken immediately before moisture addition and immediately after to see what the impact on the methane profile was. So what we see in these two graphs, so column 1 on the left, before are the closed circles, or closed shapes, open shapes are immediately after. So as you see in column 1, there's not that much difference between before and after. In general, it has overall higher methane concentration, indicating less oxidation is occurring. But in column 2, you see a difference, but it's mostly pronounced at around 10 centimeter depth, which is probably the zone of most oxidation occurring. Basically, you just see a slight increase in methane concentration after the water addition is shifting to the right. And that's expected based on previous studies that have shown the presence of water inhibits the fusion of atmospheric oxygen, which is, like I said, needed for methane oxidation to occur. No real surprises there, but I guess it's good to see that there's still some oxidation occurring relative to the control, even with moisture addition. So so I'll hurry up now. So some SEM images were taken just to show what the texture was and of the biochar and the soil. And what you see immediately is biochar has a lot of porosity, a lot of macroporosity, which is good for methanotropes in that they have sites to colonize and form a biofilm or an active community, as well as improving the gas transport through the material. So here we did a couple isotopic analyses, and what we see from this is that with a decreasing depth in the column, you get a slight enrichment or towards increasingly positive values of del13C, which is consistent with what we had hoped, and it indicates that there's some microbial fractionation of methane occurring. So that was good. And very quickly, the molecular analysis we did was a quantitative PCR of PMO-A genes, which is the methane monooxidase gene. So this is kind of like a proxy for methanotrope abundance. So in general, the biochar amended soil had a much higher PMO-A, so much more methanotropes, we assume, are present in that soil, and therefore lending that oxidation capacity. So kinetics were also done, and I'm just going to go to the results. Basically, as expected, the kinetic parameters in the biochar amended soil were much higher in terms of oxidation rate than the unamended soil. So again, that was consistent with our previous observations. So in summary, this study showed a lot of evidence for enhanced methane oxidation as a result of biochar amendments to landfill covers. We had a higher fractional conversion of methane, a higher maximum oxidation rate observed, which was greatest near the oxic zones as expected. A greater abundance of PMO-A genes, except at the lowest depth, which was not. Also increasing enrichments in 13 methane in the upper depths of biochar amended soil. And the SEM images show that there was some bacterial biofilm material present in soil pores concentrated in the oxidation zone of the biochar mini column. And yeah, so like I said, it helped increase the depths of the oxidation zone. Oxidation kinetics were enhanced, and EPS production is likely a reason for a decline in methane oxidation efficiency over time. And just to conclude, I'll be continuing some of this work with field scale and column scale experiments. So I'm going to look at a more detailed study to try and identify some of these factors that might be limiting oxidation efficiency in the long term, as well as how the effective amendment strategy on landfill cover could play a role in oxidation efficiency. So, thank you.